Hello students, Professor Lyons here, and what we're going to talk about in this video lecture is the second part of chapter 10. So in the last video lecture we talked about DNA and RNA, we talked about its structure, and we talked about DNA replication. So now we're going to go one step further and we're going to learn about the function of DNA. We're going to go from DNA to proteins and learn about how that process works. And how that process works inside of cute little critters like this guy here. This is an Atlantic puffin. You can find it in cold waters to the north, places like Iceland or Greenland or Scotland, places like that. And why I really like them is because they're awkward on land and really good in the water. So in a lot of ways they remind me like themselves. They remind me of myself, I should say. Anyway, what we're going to talk about here is a lot of what DNA is used for in our bodies and how we use DNA for those things. So I think I've kind of beaten this point into you enough by now, but anyway, as you know now by now, DNA is used as a blueprint for making proteins inside of our bodies. Proteins such as hemoglobin, such as lactase, such as ATP synthase. You know, there's tons and tons of different proteins inside of us that are really important to the functioning of our bodies. And so if it wasn't for DNA that had the information on how to make those proteins, we just couldn't exist. So what we're going to talk about is how we go from DNA to those proteins. So this is part 2 of chapter 10, going from DNA to RNA and from RNA to proteins. So first I want you to kind of be clear about the flow of information here, right? So here's a cell, right? There's the nucleus in the cell. You know that the nucleus is surrounded by a nuclear envelope that has holes in it. And the very important reason why it has holes in it is so that RNA can leave the nucleus. So the DNA is always in the nucleus. It never leaves the nucleus. The RNA does. So what we're going to see here is two different processes, right? So we start with DNA and we transcribe it into RNA in a process known as transcription, where we're essentially taking this DNA and making a copy of it in the format of RNA. That RNA then leaves the nucleus, and through a process known as translation, we take the information that is in the nucleotides, in that sequence of letters in that RNA, and we use it to make a protein. So we translate from the RNA language into the protein language. And so it's important to kind of understand the scale at which this is happening, right? So when this is going on, it's not as if your cells are transcribing all of the DNA inside of them. No, what they actually are doing is transcribing particular genes of DNA. And one gene codes for information or codes information for one specific protein. Yeah, so let's say that let's say that this stretch of DNA right here, let's say that is the ATP synthase gene. So remember, ATP synthase, that was that protein inside of mitochondria that are used for making ATP, for synthesizing ATP, which is why it's called ATP synthase. Arguably the most important protein inside of all of us, because as we know, we need a crap ton of ATP. And without ATP synthase, we can't make very much of it. So let's say this is the ATP synthase gene. So we take all of the letters in this gene, which is going to have on the order of thousands of letters of DNA, so thousands of A's and T's and C's and G's. We make a copy of it in the format of RNA, and then that RNA is used to make that specific gene, right? So we don't, so like I was saying before, we don't just take all the DNA inside of each cell and transcribe all of it into RNA. We just transcribe particular genes, right? So we take the ATP synthase gene, we make it into RNA, and then we make the specific ATP synthase protein. So that is kind of how information is gonna flow here. And so this lecture really has two parts then, because we're going to go into the transcription part, and then we'll go into the translation part. This is just showing the same thing again, but kind of blown up, right? So we have this whole long DNA molecule that has multiple genes on it. right? So each chromosome inside of you is essentially one DNA molecule, and each of those chromosomes have many, many genes on them. So hundreds to thousands of individual genes on them, right? So we take one individual gene, let's say it's the ATP synthase gene, right? So we have the letters of DNA that make up the ATP synthase gene. We transcribe it into RNA, and then sets of 3 letters of RNA then get translated into individual amino acids. So as a refresher, remember, proteins are made up of individual amino acids. Amino acids are the monomers that make up a protein, which is a polymer, right? So we make up with many, many amino acids, we can make a protein, such as ATP synthase. So let's delve a little bit more into transcription and how it actually works. So it starts with what's known as initiation. So this is when there's a particular stretch of DNA that is known as a promoter. And that is what tells RNA polymerase where to begin its work. So you probably remember that polymerase word, right? So we talked about DNA polymerase in the last video lecture, right? And so the ACE part should tell you that it's a protein. In fact, it's an enzyme, which, as you know, is a type of protein. But instead of RNA polymerase making DNA, like DNA polymerase does, RNA polymerase obviously makes RNA, right? So it is producing RNA. So RNA polymerase starts at the promoter. What it does is it pulls apart the 2 strands of DNA, it unzips it, and it goes along one of the 2 strands. It actually only reads one of the 2 strands in a DNA molecule. It doesn't read both. So we only produce 1 strand of RNA from 2 strands of DNA. So we elongate. As this RNA polymerase is going on, it is attaching individual nucleotides as it goes. So when it comes up to a T, it's going to add a U, because remember, T goes when we get to a T, we add a A, because remember, T and A, they kind of match with each other. When we find a G, we add a C, because G and C go with each other. When we find a C, we're going to add a G. So, but where this gets a little bit different from what you've learned before is if we have the nucleotide A, instead of adding a T right there, we're going to add a U, because as I was teaching you in the last video lecture, RNA doesn't use the letter T, it uses the letter U instead of it. So normally when we find an A, we would add a T to it, but T doesn't exist in the case of RNA, so we would add a U instead. So RNA polymerase is going on and reading each of these letters and adding on letters of RNA to the new growing strand of RNA. So we get to the very end, we get to what's known as a terminator sequence, and that is essentially what tells the RNA polymerase to stop. So you can think of it as a sort of finish line that tells the RNA polymerase, you know, you're done, you don't need to read any more of the letters. Okay, and as I was saying before, I wanted to be clear to you that RNA doesn't use the letter T. So anytime you find a in the DNA, you're going to add a U to the RNA. When you find a T, you're still going to add the A because A exists in RNA. It's T that doesn't exist in RNA. So there's no thymine in RNA. So if you can remember that there's no T in RNA, because the letter T isn't in the word RNA or the acronym RNA, you can remember the T doesn't go in there. So why don't you get a little bit of practice with this, right? So what I want you to do is pause the video, and I want you to take this strand of DNA and see if you can figure out what would be the RNA strand that would go along with it, right? So if you were RNA polymerase, what letters are you going to add along here? So go ahead and pause the video and write down your RNA strand, and then I'll show you what it should be. Okay, I'm assuming by now you've paused and unpaused the videos, and the video I should say, and you've written down your RNA strand. It should look like this, right? So any place you find an A, you add a U. Any place you find a C, you add a G. T, we add a A. G, we add a C. T, we add a A. So on and so forth. right? So pretty simple stuff. We're really just coding between different letters. And we just have to remember, though, when we're making RNA, we're going to use U instead of T. So that's a really important thing to remember is when you are taking the quiz or the exam that is going to include this material, make sure you read the question carefully, right? If I'm asking you to provide me with a DNA strand, you should be using T's. If I'm asking you to provide me with an RNA strand, you should be using U's, right? So make sure to pay attention to what I'm actually asking for in the question. Okay, so that was transcription, right? It's really not that complex. In a lot of ways, it's pretty similar to DNA replication. Translation gets a little bit more complicated, and that's because it is ultimately a slightly more complicated process. And why translation is a bit more complicated is because it actually is a translation of languages, right? So we're starting with the language of nucleic acids, which is in the format of A's and C's and T's and G's. In the case of RNA, the letter U. So we're starting with one language and we're translating it into the language of proteins. So the language of proteins is in the format of individual amino acids. So I taught you way back in chapter 3 that there are 20 different amino acids. In those different amino acids are like the letters of the protein language. So we're really going from one language to another, which is why translation is a slightly more complicated process. And how we essentially do it is we use 3-letter words, right? So in DNA or RNA, you can break down a sequence of DNA or RNA into what are known as codons. Oftentimes it also is known as triplets. So each set of 3 letters, say CCC or AAU or whatnot, those are what are known as codons or triplets, right? So tri meaning 3, so it's a set of 3 letters. But these 2 words are synonyms, so you'll see both of these words getting used, either codon or triplet, they mean the same thing. And so each codon or triplet, which are 3-letter words, each of those code for one specific amino acid, right? So the letters AAA, that'll code for one amino acid, and the letters TTT, that will code for one amino acid. Okay, so let's talk a little bit about what's known as the genetic code. So the genetic code is essentially the rules that are used to convert RNA into an amino acid sequence, right? So we're always going to be going from RNA to amino acids because remember remember about the information flow. We start with DNA and we use transcription to make RNA from it. That RNA then leaves the nucleus and then goes out into the cytoplasm where it can be used in order to make a sequence of amino acids, right? So amino acids are not made directly from DNA, they're made from RNA. RNA is kind of like it's the messenger between the DNA and the amino acids, which is why in a minute you're going to find out about how one type of RNA is called messenger RNA. So this is essentially what the genetic code is. So you'll see this genetic code represented in different ways. I think that the current version of the book actually shows it as a circle. I think it makes more sense shown as a graph like this, which is why I'm doing it this way. And so what you see here is you say sets of 3 letters. So remember, those are codons or triplets. And so the codon CCU is going to encode the amino acid proline, which is abbreviated pro. The codon AAU is going to code for the amino acid asparagine. What about the codon CGU? See if you can find that on the chart. What amino acid is that going to code for? Right, so you should be looking around here. So CGU codes for arginine, which is abbreviated ARG. A way to quickly find codons on this chart, because I know there's a lot of information in here, is to use this method, right? So the chart is organized this way, where the first letter of each codon is going to be represented here, right? So this whole column right here, the first letter is U for this whole column. The first letter for this whole column is C. Right along here, it's A. You can see that all of the codons in here all start with A. And so if you have, say, CGU, you know that it's going to have to be in this column right here. Then the second letter is going to be represented by the rows. So if the second letter is a G, you know it has to be in this row right here. Actually, I'm sorry, I guess I got these reversed. So these are rows, and then these up and down are columns. So say you were looking for CGU. So we take a look here. Okay, we know it has to be in this row because this is the C row. And we know it is the second letter is G, so it has to be in this column. So that shows us it's going to be like right in here, and then we can see there's CGU. So for instance, ACU, try to find that. Right, so we're going to be starting over here in this column. The second letter is C, so we know we have to be in this column, sorry, this row over here, in this column here, which shows us it's going to be on the box. And so we, in this box here, so ACU shows us that we have the amino acid threonine, or THR. Okay, so what I'm going to do is I'm going to show you a sequence of letters. And what I want you to do is pause the video when I show the letters to you, and I want you to see if you can figure out what is the sequence of amino acids that would go with that sequence of RNA. Okay, so here is your sequence of RNA letters. I want you to code that into amino acids. So go ahead and pause the video. You might have to rewind a little bit so you can see that chart. And then come back here and I'll show you what the amino acids should be. Okay, so here's what the letters should actually be. So AUG goes with met or methionine. So this is a special amino acid in that this is the start amino acid. Right, so we're always, every sequence of amino acids is always going to start with methionine, with met, because that is the start codon. UAG is one of the various, it's one of the few stop codons. So you can probably guess that when the machinery gets to UAG, it knows that that is where we need to stop. We don't need to add on any more amino acids after that. And then everything in between are just typical amino acids. So we always start with met and we always end with a stop codon. Okay, so let's talk a little bit more about how translation works. Now that you know about the genetic code and know how we go from RNA letters to amino acids. So there's kind of three really important things going on here, three important parts of the machinery. So we have mRNA from transcription. So remember I was telling you about how RNA acts as a messenger between the DNA and the amino acids. That's why that type of RNA that does that is called messenger RNA, or which is abbreviated mRNA. So we need that mRNA from transcription because that mRNA is what actually has the information in it for making the amino acids, right? That the information stored in the letters of RNA is what the machinery is going to use to produce the amino acid sequence that is correct. Another thing that we need is what's known as transfer RNA. So transfer RNA essentially acts as an interpreter. So it is what is doing the actual translating of messenger RNA codons into amino acids. So what you did in that last slide just a minute ago, where you looked at individual codons, letters of ACU and UGA and whatnot, and then figured out what amino acid would go with each of them, you did the job of transfer RNA. That's what transfer RNA actually does. And then ribosomes, this is where it all actually happens. So ribosomes, you may remember back from chapter 4, that is where proteins are made. And so now that should make sense because now we're going to talk about how ribosomes are actually involved in this process. In ribosomes, as we talked about, they can either be embedded within membranes. So they can be on endoplasmic reticulum, they can be on the surface of the nucleus, or they can just be floating around within the cytoplasm inside of the cell. Okay, so let's talk a little bit more about the transfer RNA. We don't need to talk anymore about messenger RNA because you already really know all about that because we talked about how we make it in transcription. But transfer RNA is a very special type of RNA. And what is special about it is that it has two different attachment sites. So it has a set of 3 letters that are exposed here that are known as the anticodon. And so those 3 letters are going to match up with specific letters of messenger RNA. So if the messenger RNA reads, if it reads UUU, only the transfer RNA that has AAA right here can actually bind to it. Because remember, A is going to go with U. Or say we have a set of messenger RNA that is CCC. The transfer RNA that has GGG is the only one that is going to be able to line up with it and bind to that specific messenger RNA. So this is what's known as the anticodon because it is the opposite of the codons of messenger RNA. And then on the other side, we have the amino acid attachment site. Right, so this is where those various amino acids like methionine or tryptophan or arginine, this is where they are going to bond onto. And so one really important thing about all of this is that transfer RNAs, each one is specific to the anticodon, so it has a specific set of letters here, and it will only bond with one particular amino acid. And that's really important because if every transfer RNA could bind to every one of those 20 amino acids, then you wouldn't end up with the correct sequence of amino acids. You would just end up with just a random list of amino acids. It's important that these things match up with each other appropriately. It's kind of in the same way when you translate a language. You know, say you had a language translation dictionary. When you look at a translation, dictionary, you put in one word, and only one other word should then come out on the other side in that other language. If you put in a word and it just gave you a random, assortment of words, then things wouldn't work out so well. Then you wouldn't be able to translate from one language to the next. So like translating between languages, we really have to go from one specific thing, the anticodon, to one specific amino acid. Otherwise, this whole system just doesn't work. Just like a translation dictionary wouldn't work if you put in one word and we're just given a random word in that other language. Okay, so ribosomes are really important to all of this as well. And ribosomes are actually made of 2 different parts. So there's what's known as a small subunit of a ribosome, and there's what's known as a large subunit of a ribosome. And what happens is there's 2 different binding sites within that ribosome, and that's where the tRNA actually goes. So the tRNA binds to within these 2 different sites on the large subunit, whereas the messenger RNA binds to the small subunit. And so this is then showing where everything fits in place. So here's the messenger RNA in red, here's the transfer RNA there in green, and these purple block things, those are the individual amino acids. Okay, so let's talk about the actual process of translation. So we start with messenger RNA that came from transcription, and it binds to a small ribosome subunit. And after it does that, then the first tRNA that has methionine, so that's the beginning amino acid, it's going to bind to the start codon, to the letters AUG. And so remember, like I was saying before, if we have a set of 3 letters like AUG in the messenger RNA, only the tRNA that has the opposite of that has the correct anticodon can bond there. So A bonds with U, U bonds with A, and G bonds with C. So these 3 letters here are a match for AUG. So this transfer RNA here can attach right there. And that brings in this first amino acid, methionine. Once that happens, then the large subunit can attach, and now we essentially have all of the necessary parts to make this whole process happen. So this is the initiation phase of translation. The next part uh, of translation is what's known as elongation. And so elongation kind of occurs through three different parts of a cycle. So we've got our transfer RNA, we've got our messenger RNA, we've got our two ribosome subunits, and we've got a growing strand of amino acids. So what happens is a new transfer RNA with its amino acid comes in and it binds to the A site of the ribosome. So it binds right there. And then what you'll have is two transfer RNAs that are right next to each other, meaning that there are two amino acids right next to each other. What happens is those two amino acids bind to each other. And when they do, this circle amino acid detaches from this transfer RNA that was here at the start of this process. So it detaches from there. Once that transfer RNA is no longer attached to the amino acids, its job is done. So what it does is it pops off of the large ribosome subunit. So it's now gone. It's going to go attach to another amino acid and repeat the process again. So now we have this empty space over here. And what essentially happens is this transfer RNA, it translocates over to this site. So it goes from the A site to the P site. And essentially the whole messenger RNA shifts to the left. So now this transfer RNA is over here and the messenger RNA is shifted over by 3 letters. So now we're back at the beginning. So we have a transfer RNA here. This site here is empty. A new transfer RNA can come in. The amino acids bond. This transfer RNA detaches from the amino acids and then it pops off and leaves. And the whole thing translocates over one place. So I know that it's kind of a hard process to describe just with a flat image, which is why I have I put a link to a video that describes it with some nice graphics that you can take a look at. So if this is still kind of confusing to you, then take a look at that video. So then the very last part of translation is when this machinery reaches a stop codon. So remember, there's a few different stop codons. What essentially happens when you reach one of those is a different type of, a special type of transfer RNA comes in and binds there and makes all of these various parts come apart. So the two ribosome subunits come apart, the messenger RNA detaches, and the amino acid sequence, the set of amino acids detaches from the transfer RNA and goes away. And so that's basically how translation works. So remember, back at the very beginning, we started with DNA. That DNA through transcription was made into RNA, which you see here in red. And then that RNA, through the process of translation, is used to make a sequence of amino acids. And a sequence of amino acids is essentially a protein. So that's how we go from DNA to a protein. So that's how we go from, say, a gene in your DNA sequence, say the ATP synthase gene. That's how we actually then make the ATP synthase protein. Okay, so one last thing that I want to talk about in the context of DNA replication and transcription and translation is what are known as mutations. So mutations are changes in the sequence of nucleotides. So it's any alteration to those letters. So a good example of this happens with the hemoglobin protein. Right, so hemoglobin, as you remember, is the protein that is on the surface of your blood cells. That protein is what binds to oxygen and allows your blood to carry oxygen throughout. And what most people have in their hemoglobin gene is in the middle here, we have this set of CTT. When we then transcribe that into messenger RNA, we end up with GAA. And when we translate that into a protein, we end up with a protein glutamine. And so what that results in is a hemoglobin gene that causes a red blood cell to have its typical kind of round donut shape. So, but what happened thousands and thousands of years ago is there was a mutation that still many people carry today, where the T mutated into an A. So when we have CAT, now we end up with the messenger RNA letter GUA. And when we have the gene, when we have the amino acid here, we end up with valine rather than glutamine. And what that results in is a red blood cell that has this kind of crescent, kind of sickle shape. So I think probably a lot of you are picking up on the fact that this is what causes a person to have sickle cell anemia. If they inherit from their parents an A in this spot rather than a T, then they will end up with red blood cells that have this sickle shape. In red blood cells that have this sickle shape, comes with, a lot of problems. Specifically, there's two really major problems. One problem is tiredness, because then the red blood cells can't carry as much oxygen. If you can't get as much oxygen to your tissues, then you can't make as much ATP, which means that you're going to be, prone to tiredness. So that's one major issue. Another issue is with clotting. So the red blood cells that have this shape are more likely to get stuck inside of veins and capillaries. So that can cause really excruciating pain. But even more serious than that, it can cause a heart attack or it can cause a stroke. Because if you end up with one of these blood cells, clotting the carotid artery that goes up to your brain, that's what causes a stroke. Because then there's not adequate blood flow to the brain, which can cause permanent brain damage or death. So having this copy of the hemoglobin gene can be very dangerous for those reasons. So then you might be wondering, why is it that this mutation from T to A, why did it occur, and why is it that it still persists today? You would think that a long time ago, this copy or this version of hemoglobin would have been, would have gone away, because people that had it would have passed away and not had offspring. So the reason why it still persists today is because this mutation from a T to A, it occurred in an area where there's a lot of malaria. And actually having red blood cells that has this particular shape makes a person less prone to malaria. So malaria is, of course, really, really dangerous. And malaria, can kill you at a very, very young age. Whereas people with sickle cell anemia, they can still, grow to adult age and can oftentimes, live, to be quite old, and can have offspring. But if you get malaria when you're a kid and you die, then obviously you're not going to have any kids and pass your genes, onto your kids. So that's why it's this version of the gene is mostly found in African populations, because this mutation from a T to a A, occurred amongst African populations, many years ago, and still persists today because it is still an effective barrier against malaria. Okay, so let's talk about how mutations occur. So mutations are caused by one of two things, well, one of three things really. So there are substitutions, where we exchange one letter for another. Say we exchange a G for an A. There are deletions, where we remove one of the letters, and there are insertions where we add another letter. And so I should say that in general, for these things to happen, it's actually quite rare. So when DNA polymerase is doing its job of replicating DNA, it in general does an incredibly good job. It is super, super precise. It will read, millions and millions and millions of letters without making a single mistake. But once every while, once every few million letters, it might make one individual mistake. And so if that mistake occurs when your body is making a sperm cell, now that mutation is going to be passed on to your offspring. or if that mutation occurs when your body is making an egg cell and that mutation goes in that egg, then that mutation is then passed on to your offspring. And that means that every single cell inside of your offspring's body would then have that mutation. If this substitution or deletion or insertion, occurred, say, in a cell at the tip of your finger, you wouldn't pass that on to your offspring. It really needs to be a mutation, in the sperm cell or in a sperm cell or in an egg cell. But what that cell at the tip of your finger with that mutation in it can then cause is cancer. Because what causes cancer to occur is when cells accumulate mutations, and those mutations cause the cells to start dividing rapidly. And so mutations to, cells in your body that aren't sperm cells or egg cells can still be quite problematic because they can lead to cancers. So let's talk about what substitutions, deletions, and insertions, what they can lead to. So substitutions can lead to a couple different things. So they can lead to what are known as missense mutations. So this is when we exchange one amino acid for another amino acid. Right? So let's say we were supposed to have UUU, but let's say there was a mutation that resulted in UCU. Now we have exchanged one amino acid for a different amino acid. So that is the case of sickle cell anemia, right? We exchange just one amino acid for a different amino acid. Nonsense mutations are far worse. Nonsense mutations are when we exchange one amino acid for a stop codon, right? So let's say we were supposed to have the letters UAC, but instead we have a mutation that results in UAA. Now we end up with a stop codon where there isn't supposed to be a stop codon, meaning that all of the other amino acids that were supposed to go after that stop codon now aren't going to be put there. So now the protein that results from it is not going to be complete and it's not going to be able to function. And if it's an incredibly important protein like hemoglobin or ATP synthase, that is going to, cause a person that is just never going to even be born. Because you can't survive without ATP synthase, you can't survive without hemoglobin. So stop codons, if they occur on really important, genes, then they're always going to be lethal. So they're really, really bad. Silent mutations are kind of an interesting one. So a silent mutation is when we exchange one letter of DNA for another letter, but we don't actually end up with any amino acid change. We end up with the same amino acid. So take a look at this part of the genetic code. See if you can figure out why that is. How could it be that we would exchange one letter for another, but don't actually see a change in the amino acid? Right, so some of you might be noticing that for different amino acids, there are multiple sets of codons that can produce them. So for instance, with the amino acid proline, you can produce proline with CCU, CCC, CCA, or CCG. So say you were supposed to have CCU in one particular part of your RNA sequence. If there's a mutation, so now instead it's CCC, you're still going to produce proline. There's not going to be any change in the amino acid sequence with even though there was a change in the RNA sequence. So silent mutations have no effect because you still have the correct amino acid, and so you still end up with the correct functioning protein. So that's what substitutions lead to. Insertions and deletions, they lead to what are known as frameshift mutations. And what that results in is typically the correct number of amino acids, but all of the amino acids are wrong. So let's consider why that is. Let's say you had a sentence like this: the dog ran and ate. If you were to cut the letter H out of it and then shift all of the letters over one place, you would end up with a different sentence, right? If we cut that H out, now the E moves to that spot, the D moves to this spot, the O moves to this spot, and what you end up with is Ted Ogre Anna Nada Tay, right? You don't end up with the same thing. And so the same thing occurs with letters of RNA. If you cut out one letter of RNA, everything shifts over, right? So if you cut out a letter, everything shifts over to the left. And so then we end up with different codons. If you add in a letter, everything shifts over to the right. And again, you end up with different codons. So insertions and deletions are really quite bad because all of these amino acids are wrong. So anytime there's an insertion or a deletion, it produces a protein that is not going to be functioning. And if it's a really important protein like ATP synthase or hemoglobin, it's going to be lethal. Okay, so let's talk about how mutations occur, right? So how do we end up with substitutions and insertions and deletions? So like I was saying, mutations occur spontaneously by DNA replication. So the machinery, in this case DNA polymerase, does a really, really good job. So in general, it can read millions of letters and only make, one or two mistakes. It's really good at what it does. However, every now and then it does make a mistake. So it can just happen just spontaneously. However, there are things in the environment around us that can increase the likelihood that mutations happen, right? So these are what are known as mutagens. And so things like nuclear radiation, from like nuclear power plant mishaps or from nuclear bombs, so think of like Chernobyl, in Ukraine, or like Fukushima in Japan, where you had nuclear power plants that malfunctioned and caused releases of radiation. That radiation release, causes mutations, and it's really, really harmful to the health of the people that come in contact with it. The same with X-rays. You probably know from experience, when you go to a doctor or to a dentist and they take X-rays of your teeth or of a bone, you know that the X-ray technician goes into another room when they do so. And that's because, they're taking tons and tons of X-rays every day. X-rays are mutagenic. So they're doing it for their own health so that they don't get lethal doses of X-ray radiation that could cause lots of mutations. And that's why they put on you a lead vest so that they can try to protect your really valuable organs inside. UV light is another mutagen, right? So if you expose yourself to too much UV light, skin cells can accumulate mutations. And if those mutations cause those skin cells to start growing really rapidly, that's what causes skin cancer. So in actuality, another word for a mutagen is a carcinogen, right? So carcinogens are things that increase the likelihood that your cells get mutations in their DNA. And those mutations can sometimes cause those cells to start growing and dividing really rapidly. So we'll talk about that a little bit more later when we get into chapter 8 and we talk about how cells grow and divide. All right, so that's all I've got to tell you for now about DNA and about DNA replication and how we take DNA and we make proteins from it. So in the next chapter, we're going to talk a little bit more about some of the interesting technologies that exist for manipulating DNA and for using DNA to our own advantage for making various products or for forensics.